I just wanted to run through viruses with you. So viruses are really, really small. They're about 50 times smaller than bacteria. They're only visible with an electron microscope. You can't see them with a light microscope. You need an electron microscope to see them, okay? Examples of diseases, human diseases that are caused by viruses: measles, chickenpox, rubella, mumps. So the MMR is a vaccine that we all get as babies that prevent these viral diseases or prevents these viral infections. So remember what a vaccine is now from the previous chapter. A vaccine is an example of an artificial active immunity. Why are vaccines active immunity? Well, because you make the antibody yourself. So remember, a vaccine is like a treated dose of the virus. So it's treated in such a way that the virus can't actually replicate inside your body, but it does cause your immune system, your B lymphocytes, to make antibodies and memory B cells that will remain, so that if you do get infected with measles, mumps, or rubella again, you've got the B memory cells there on patrol, if you like, going around your body, and if they see the virus, then they'll mount an attack really quickly, so they'll produce lots of antibodies really fast. So that's the idea of the vaccine. Herpes is another example. There's some other examples of viral diseases. And you need also to know some examples of animal ones. So like foot and mouth, rabies, cowpox, myxomatosis, they would all be examples of viral animal diseases. Obviously, the coronavirus or COVID-19 is a typical or topical one now at the moment as well. Okay? Now, swine flu, so there's loads. Now, there are different shapes of viruses. So viruses can be round, rod, complex, polyhedral. So they're the shapes of viruses, okay? And they're just some examples of those. A bacteriophage is a virus that infects a bacteria. So there's been lots of research in recent years into the use of bacteriophages to try to combat antibiotic resistant bacteria. So superbugs, bacteria that we don't have an antibiotic to kill. Those kind of bacteria, there's been research into using these viruses to infect those bacteria and causing their destruction, if you like. So bacteriophages, again, are viruses that infect bacteria. Okay, now, and there you can see there's more shapes. Now, the basic structure of a virus, it's actually quite a straightforward entity, I suppose. It's not cellular, okay? So it's not composed of cells. It is essentially a protein coat called a capsid with then either RNA or DNA. So just one type of nucleic acid, RNA or DNA. Now, some viruses, like they're saying the coronavirus is an example of one that has an outer membrane, normally made of some sort of a lipid component. So maybe a glycoproteins, that there's the lipid and protein attached or, composed of this outer membrane. That's why they're talking about washing your hands with soap to try and disrupt that outer membrane of the virus. SARS is another example of a virus that's been, in recent years. And there you can see, look, the basic structure of the virus. So it's a capsid coat, so it's like a protein coat, and then you've got your DNA or your RNA. No organelles, no ribosomes, no mitochondria, okay, no cytoplasm. So viruses have to have a host in order to be able to undergo metabolic reactions. They don't carry out any metabolic reactions themselves because they don't have any organelles. Now, there is evidence that they can actually survive on hard surfaces for a few days, but after that then they can't, because they need a living host in order to be able to replicate, in order to be able to survive. Now, that leads into the argument then, scientists are, they don't know, they're not a part of the five kingdoms. Do you remember the five kingdoms we would have spoken about? Monera, fungi, protista, which are the amoeba, plants and animals. So the viruses are not included in those living kingdoms. So do you remember we would have spoken about Eron, the very beginning of fifth there? So the characteristics of life. So if you go through those, Eron, so E, excretion, viruses don't excrete, right? Reproduction, they actually don't reproduce either. They replicate because they have to have a living host for replication. Okay, respiration, no, they can't undergo respiration because they don't have any mitochondria. Organization means that there's basic cells organized into tissues and then tissues into organs. No, they don't have cells, so they don't they don't have any organization like that. And then nutrition, no, they don't. They take over the actual living host, and they obtain they use their organelles. So no, they don't have any mode of nutrition either. So there is an argument here. So the living factors, yes, they do have RNA, they do have DNA, they do have a protein coat, and they can replicate, but only once they're inside a living host. So we don't say they can reproduce by themselves, because they can't. Non-living factors, then, they only have one type of genetic information. They only have the DNA or the RNA. They're not composed of cells. They cannot reproduce themselves, and they don't have any kind of organelles. How do we control viruses? Well, you've heard an awful lot in the news at the moment about trying to develop a vaccine against the coronavirus. So vaccines are our most effective method of control. So Virax, is a sort of an antiviral cream that's used to treat the herpes virus. So herpes might cause a cold sore or something. So sometimes if you feel you have a cold sore coming along, you might put on some Virax on that. Interferons, we spoke about these defense proteins in the general defense system. So these are substances that are made by viral infected cells that help to protect the cells, and antiviral drugs then. Now, you need to know about the replication of a virus. So not the reproduction now, it's replication. So there are 5 stages. Okay, attachment, entry, synthesis, assembly, and release. And try and think of a sentence, AESAR. I don't know, come up with something there. Now, I'm going to use a bacteriophage as an example. So remember, a bacteriophage is a virus that infects a bacterial cell, but the virus would infect human cells in the same way, okay? So first, the attachment is where the proteins that you see sticking out of the surface of a virus, those proteins will attach to cell proteins. So they attach onto the cell proteins, and generally they're known to be specific, so that a particular virus would attach to a particular cell, okay? Then what happens is that they inject in the RNA. So attachment and entry, so entry of the RNA, so the RNA is injected into the cell. Sometimes the whole virus goes into the cell, and then the actual protein coat will be digested by the enzymes in the cell. But in this instance, for bacteriophages, it's normally just the RNA that's injected in. Okay. The third stage then is where the hosts, so the bacteria cell or the human cell's host DNA is kind of made inactive, I suppose. And then the viral nucleic acid takes over. So the RNA that was injected in takes over the cell's organelle. So it takes over the ribosomes, and it makes protein for the virus itself. It takes over the mitochondria to produce energy, and it makes copies of its own RNA. So the viral RNA is copied, not the host's DNA. That's made inactive. And the viral RNA is copied inside in the host cell. And then assembly means that new viruses are put together. So new copies of the RNA and new protein coats are assembled together to make new viruses. And then the final stage is release, where the bacteria cell or the human cell will burst, if you like, and release thousands of viruses that will then infect new cells. Okay, so you need those stages: attachment, entry, synthesis, assembly, and release. Now, you need 2 benefits of viruses. So the first one is vectors. And when we were doing genetic engineering last year, we spoke about vectors and viruses, VV. So vectors transfer genes from one organism into another organism. Okay, so viruses are used for that. And also the control of infection. So I spoke to you earlier there about bacteriophages and how we could use those viruses to try and destroy bacteria that are resistant to antibiotics. Okay, so that would be important. Okay, now the disadvantages then, of course, would be that they cause human disease. So you can see there AIDS and the coronavirus at the moment causing human disease, and also plant diseases. Okay, so you need to know a few examples of plant diseases as well that viruses would cause. So I thought I had a slide there, but things like tobacco mosaic disease, okay, tomato mosaic disease, potato mosaic disease, those kind of plant diseases are responsible, are caused by a virus as well. Don't seem to have it. Okay, now your revision book there on page 77 does out a full page on viruses. There isn't much to it really, and I'll put this PowerPoint on Edmodo.